This is a very interesting topics, as the name implies, epigenetics, and I have included microRNAs also. I will tell you why. So, in the human genome, we learnt there are about 20 to 30,000 genes, and we also know that all the tissues have the same genetic information. A liver tissue is a liver tissue because it expresses a certain set of genes, and same thing with kidney tissue or any other tissue, because they have specific gene expression, tissue specific gene expression. So, it helps us to identify a particular tissue. So, gene expression is very important. Now, in with respect to cancer biology, all of you know that the cancer patients have activated oncogenes, at the same time inactivated tumor suppressor genes. How the tumor suppressor genes are inactivated? It can be a deletion mutation, can be something like that, but more importantly, epigenetic silencing of the tumor suppressor gene is the major mechanism than mutations. This accounts for the loss of expression of tumor suppressor genes in cancer. So, the major mechanism by which tumor suppressor genes lose their activity is by epigenetic silencing, not simply by mutation. Mutation is also there, but the major reason is this. That is the importance of epigenetics and cancer basically. Now, we have to know what is epigenetics first of all. Genetics cannot explain all the differences between phenotype and genotype. For example, in differentiation, it cannot account for the drastic changes. You take all of us are born between, like from the fusion of sperm and ovum, which we ultimately call zygote, right. There are only two cells that fused into a single cell, which we call as zygote. When you look at the zygote, it looks like just a mass of cells, nothing is visible, right. It looks like a very crude structure. Whereas, when the very same cell differentiates into a fetus, you start seeing eyes, nose, all structures are visible, some functions are visible. So, there is a drastic change. You cannot simply say that undifferentiated to differentiated tissue happens because of genetics only, genetic information only. There may be something above it. Epigenetics literally means, in Greek, epi means above. Above that there is something else, or in other words, apart from that, or in addition to that, there is something else. What is that something else? So, people thought that there is something that tells that this tissue has to, this set of genes have to undergo certain fate, that is called crisis. So, that determines their fate. Hence, it was thought undifferentiated cells undergo a crisis that determines their fate. So, this crisis was not dependent on genes alone. So, it was thought that this happens in addition to genes, that is what is called epigenetics. Now, we will just see what are the epigenetic changes, who discovered this. In 1939, Conrad Waddington is credited for coining the term epigenetics, as I said, literally meaning above genetics. If you want to give a textbook definition, the epigenetics means the study of mitotically and meiotically heritable changes in gene expression that occur without a change in DNA sequence. DNA sequence is not changed, but expression changes. That is the point. DNA sequence is not changed, but expression is controlled, regulated by certain changes which are inherited. So, that could have happened through mitosis or meiosis. So, that is the idea. All of us are kind of familiar, many correlate epigenetic changes to DNA methylation, and many people think nothing more than that, but that is not correct. There are also changes in the chromatin. Histone also undergoes a lot of changes, and in addition now the non-coding RNAs also. So, there are three things. One is DNA, changes in the DNA, changes in the histone proteins, the chromatin. Chromatin, you know, is a combination of histone and DNA. The moment it combines with that and is kept in a compacted way, you know that you get the structure for the chromosome. What you see chromosome from the electron microscope and the typical structures that you see immediately shows it is a compact structure, and it is mixed with the proteins also, that the so called histones. All of you know about this. So, in addition the non-coding RNAs, because they can control expression ultimately. Non-coding RNAs, some of the microRNAs for example, they can also control. Again, that does not change the DNA sequence, correct? It only changes the expression. So, that is why we are including microRNAs also into. So, three things: DNA, methylation and other changes, chromatin changes and microRNAs. They contribute to what are called epigenetic changes. We are talking about cancer biology. So, tumors exhibit an overall loss in DNA methylation, but also show specific patterns of hypermethylation in certain promoters. The promoters, you know, will have the methylation sites, right? The promoters can be methylated or hypermethylated, or there may be loss of methylation in that promoter and so on. These consequently make changes in the gene expression. So, that has to be understood. Now, all of us know that DNA is packaged into nucleosomes characterized by DNA chains, approximately 146 base pairs wrapped around histone octamers. You know, there is H1, H2, H3, H4 histones and all that. I hope you know the octamer structure and all that. Histones are basically small proteins, basic proteins, 11 to 21 kilodalton, that bind non-covalently with acidic DNA to form, because this is basic, that is acidic. So, there can be a binding, although it is non-covalent. So, to form nucleosomes, that is what this combination you call as nucleosomes. And of course, chromatin is an overall structure, the whole thing, which also has this histones as well as the DNA. Octamers are formed by H2A, H2B and H3 and H4. They are present as dimers, so you count it as 8, because there are 4 histones, they are dimers, so it is 8 octamers. This is basics. So, this is how it looks like. Suppose this let us say a chromosome, unreplicated chromosome. The chromatin is, you know, the beads on a coil, we say. So, the beads are contributed by the so called histone octamers, and so there are some protrusions which we call as tails, both at the N terminal and the C terminal usually, and they can undergo post translational changes. What are the post translational changes? Here we are talking about histones, methylation, phosphorylation, acetylation. So, these are some prominent changes. There are some additional changes, but these are the most important ones. The portions of the DNA bound with histones and other proteins should be exposed for facilitating transcription. Whenever there is a tight binding of histones with DNA, the chromosomes are said to be somewhat condensed. Okay. So, in that case transcription may not happen, because for transcription what is the requirement? The transcription factor, which is a protein, has to go and bind the DNA. If DNA is surrounded by proteins and covered up and there is no space to enter, then how the transcription will take place? So, it has to open up. So, that is what is important, what we call as the nucleosomes. If they are packaged into compact aggregates, then they silence the transcription, and that is called closed chromatin. In contrast, if they are packed loosely, then it is called open chromatin. This is sometimes called like valency, it is called bivalent. Sometimes people call that chromatin as bivalent because it can remain either in closed conformation or in open conformation. That is what they say. The post-translational modifications are on key amino acid residues of the histone is are the important ones which control the expression ultimately. So, this is sometimes called histone code. The histone code is very important for the regulation of gene expression. So, they basically histones, just they are not some partners or structural partners that will help in DNA packaging, because in DNA is packaged so compactly that in one cell, if you stretch the DNA, it can go to something like 5 meters, and if you stretch all the DNA that is present in the human body, then it can go anywhere, like even to the moon, something like that, if you keep them, if you imagine like that. But anyway, what it means is that there is a compact structure. DNA along with these proteins is held in a very compact situation. But that is not the only thing. There are several modifications, covalent modifications of histones, like I told you, post-translational modifications. The important ones are, usually they happen in the amino acid lysine, because lysine has these amino groups, extra amino groups, right. How many amino groups are there in lysine? Anybody knows the lysine structure? Lysine is a basic amino acid, and usually amino acid means it will have one amino group and one carboxyl group, correct. But lysine has extra amino acids. How many? Only one extra? No. 2 extra, that means it makes it 3 amino groups, right. So, 3 can be there. I will tell you why we are talking about it shortly. So, then methylation, again this is another confusion with students. Whenever I ask in the interview methylation, they all know DNA can be methylated. Immediate confusion is whether proteins can be methylated. You are all forgetting that there is an amino acid called methionine, right. So, methylation is possible, right. Also lysine can be again methylated. There are several proteins which can be methylated. So, methylation, lysine, arginine I have written here. Phosphorylation is another thing. Again phosphorylation can happen in serine and threonines. Tyrosine also can be phosphorylated, but here we are talking about these histone modifications, usually serine and threonines. Then there is ribosylation, ADP ribosylation of glutamic acids. Then what is called ubiquitination. There is also a new modification called sumoylation. All these things happen in this ubiquitination, and sumoylation also happen in lysine residue. So, lysine is a very potential amino acid which can undergo several changes, not simply methylation or something like that, acetylation, methylation, sumoylation, ubiquitination. So many changes can happen in lysine. This is an important point that people do not really pay attention to that. Similarly, methylation, you think only DNA will be methylated, RNA also can be methylated. Do you know that? Many people do not pay attention to this because they are not showing any visible or very important changes, something like that. So, people think that it cannot happen. You have to keep in mind. So, status of methylation and acetylation on specific lysine residues within the tails of the nucleosome core histones is known to have a crucial role in regulating chromatin structure as well as gene expression, because it has to kind of, when there is acetylation and all, it kind of opens up, and that helps in transcription. That is why whenever you have this acetylation and methylation and all that on lysine, here we are talking about proteins, do not get confused with DNA, then it helps in transcription because the structure opens up. Acetylation of some residues, for example, this is also very important. Lysine at the 9th position of histone H3, that is what they write, H3K9, by what is the enzyme that acetylates? It is called HAT, histone acetyl transferase. So, usually when you get this acetylation, it is transcriptionally active. That is usually, exceptions are there. See, it all depends on what is getting transcribed. If for example, if because of this transcription, let us say the gene that is transcribed is a deacetylase or something that inhibits transcription, then the transcription may be inhibited. We are saying it is transcriptionally active. Yes, that we are saying because transcription takes place. So, we say it is transcriptionally active, but out of that transcription what happens? It may be transcribing an inhibitor gene. So, ultimately transcription can be inhibited also. It is context dependent, but generally it is increased transcription. So, deacetylation by, this is another set of enzymes, HDACs, histone deacetylases, usually signifies transcriptional repression. So, I have worded that very clearly. Usually acetylation results in transcriptionally active regions, right, enhancement of transcriptionally active regions. Similarly, deacetylation means transcriptional repression. There are exceptions, that is what I wanted you to remember. Histone tail methylation can be activating or inactivating depending on the site. So, when I say H3K4 methylation means transcriptionally active areas, while H3K9, what this means? H3 means histone 3, K4 means, what is the meaning of that? Lysine. K means lysine here, right, K9, that position of that lysine. K27 means at the 27th position. Methylation suggests transcriptionally repressed regions. See, 27 means it is repression, whereas K9 means it is activation. Sorry, K9 and K9 and 27 it is repression, whereas K4 is transcriptionally active. So, it depends on where it is getting methylated. Histone methyl transferases are the enzymes which contribute for this methylation, and this is sometimes known as a methylation mark, and the enzymes are enzymes are known to be writers of the code, because this is called histone code, I told you, marks. So, it is called writers. The demethylases, since they remove this mark, they are called erasers of the code. Sometimes this terminology is used. It is not that it is always used, but people use this kind of technology, terms to distinguish it from gene coding. Codons are different, right. So, this is at the protein level we are talking. So, it's called histone code and writers and erasers of the code. to denote those enzymes. Well-known epigenetic modification in humans is usually in the DNA, all of us know, especially within the dinucleotide CpG, where C means cytosine, G means guanosine, and P means what? Phosphodiester bond. It links. See, the nucleotides are linked. You know the phosphate sugar and base backbone. So, CpG refers to both methylated and unmethylated dinucleotide. Generally, when you say CpG, it simply means C and G linked by a phosphodiester bond, right. P refers to the phosphate moiety that connects the deoxycytidine, because we are talking about DNA, we are saying deoxycytidine and deoxyguanosine. So, CpGs occur in the genome in a lower frequency because methylated cytosines can spontaneously deaminate to form thymine. For this, you should know the structure of thymine and cytosine. So, if you deaminate that methylated cytosine, then you get thymine. That is a very important thing. DNA repair machinery actually does not recognize this, leading to the accumulation of CT mutations during evolution. This is very important, and 99% of the genome is CpG depleted and only 1% is composed of discrete regions that have a high GC content and CpG content. So, this is also an observation, it is very important. DNA methylation occurs along with other epigenetic modifications. As I told you, it is not just this DNA methylation alone contributes to the epigenetic changes. Histone modifications, microRNAs, they also contribute. So, along with other changes, is closely connected with the formation of nuclease resistant chromatin, because the chromatin may open up or closed. So, those changes can be affected depending upon these methylation and other changes that happen. Methyl CpG binding proteins, yes, there are proteins which specifically bind the methyl CpG, and DNA methyl transferases, so called DNMTs, DNA methyl transferases, are all associated with histone deacetylases and histone methyl transferases. What it means is, we are talking about different enzymes. There are methylases, demethylases, there are acetylases, deacetylases. Like I told you, for everything there is something opposite. For our understanding, we are studying them separately, but in biology, in the cells, they are together. The enzyme complexes are there, they are all together, many proteins are together. So, it is very important that they are together and they bind certain proteins, like here I am saying methyl CpG binding proteins, they will also bind DNA methyl transferases. So, like that they will be made into a complex. The CpG rich regions are sometimes called CpG islands. This is also a very important term, where you have very low CpG density close to CpG islands, then you call it as shores, just like shores and islands. So, people have this terminology also. In cancer cells, transcriptional silencing of tumor suppressor genes occurs by CpG island promoter hypermethylation. This is a very important thing. Remember, I told you tumor suppressor genes are silenced mainly by epigenetic changes, right. This is the most important epigenetic change. What is the epigenetic change? Promoter hypermethylation. For a particular gene, there may be a methylation at particular points If it is hypermethylated, so many methyl groups are, so many places you have methylation, large number of methyl groups are there in that place, then you have gene silencing. So, tumor suppressor genes can be silenced like that. We will talk about those details. Just a few minutes back I told you, this is a structure of cytosine. I hope you know the structures of all these pyrimidines and purines and all that. This is what, purine or pyrimidine? Cytosine? Pyrimidine, yeah. So, you have this amino group. There are generally, methylation, demethylation, amination and deamination will happen, and then oxidation. When these things happen together, you call that as oxidative deamination. So, these are the changes, biochemical changes that are very important: oxidation, reduction, amination, deamination. When oxidation and deamination happen together, then you call it as oxidative deamination. So, here you can see cytosine structure. Once you oxidize that and then remove the amino group, that is just now I told you oxidative deamination. So, then you get uracil. So, uracil is formed upon oxidative deamination of cytosine. Similarly, this will be considered a mutation and hence will be removed by DNA repair system. This is a very important statement what I have given. That is why we have thymine but not uracil in DNA. If somebody asks you why thymine is present in DNA but not uracil, this is the reason. Because uracil is formed by oxidative deamination, and so this will be considered as a mutation by the DNA repair system and will be removed. You can ask, sir, it is present in RNA, it is not removing, because it is recognizing only DNA, right. So, do not get confused because of that. So, we are talking about DNA. In DNA, if this change happens, then that is removed. Yeah. So, that is the idea. Methylated cytosine upon that also is written here, cytosine. If it is methylated, you get 5-methyl cytosine. That again upon deamination can form thymine. What I want you to understand is this cytosine, uracil, thymine, they are all very similar molecules. Just by certain changes in the amino group and methyl group, they can be converted into one another. So, that is a very important point. So, methylated cytosine upon oxidative deamination changes into thymine. Although this is basic, I just wanted you to pay attention to that, because this is very important for understanding. Many interesting things are there about methylation. One of the important things is, have you ever thought about your methylation status in the DNA and your diet? Suppose you eat something, your methylation changes. Can you believe that? It happens. Many people, I had a very interesting incident wherein I went for a PhD viva in Indian Institute of Science. Student was brilliant and they worked on epigenetics only. But when I asked the student whether there is any connection with the diet and methylation, she could not answer. I was surprised actually, because this folate, whenever there is methylation, you should think who donates that methyl group and who facilitates that. Methylation comes from the so called adenosyl methionine, and this is what biochemistry teaches us. Once we know the biochemical reactions, we know where from it is formed. So, we will be very clear. The diet, folate is a vitamin. So, vitamin means necessarily you have to take it in the diet, right. Vitamins, how do you get ultimately? From the diet, right. So, you get dihydrofolate and then tetrahydrofolate and that forms these reactions. Another vitamin is also required, B12. So, you get homocysteine which gets converted to methionine. You require ATP and then only you get S-adenosyl methionine, which is the methyl donor. And what facilitates that methylation? Methyl transferase. The donor is S-adenosyl methionine, it is converted to S-adenosyl homocysteine and you require this DNA methyl transferase. So, for methylation the most important point is where the methyl group came from. Who is the methyl donor? S-adenosyl methionine. Who facilitates that? Methyl transferase, DNMT as we call short form. So, this is very important, and then of course, why we are talking about it? CpG island, you see unmethylated CpG can get methylated now. What residue gets methylated? Cytosine. Now, if I ask you, this is another interesting question that we ask in the PhD interviews also, what gets methylated in the DNA? Prompt reply, cytosine. What else can get methylated? Other residues are there, ATCG is there. Then the answer is silence. No answer. People do not say. Adenosyl methionine. So, adenine also can get methylated, right? Yes or no? Yes. So, even guanosine can get methylated. So, it can be methylated, but what is important is cytosine, of course, because it is involved in gene expression. So, do not get confused. Similarly, methylation can happen in proteins also. These are all the usual confusions for the students, because once you study the textbooks typically give what is important and what are the functional consequences, but it requires some deeper thinking to understand what is happening around. So, this is a very important one. Diet also has a role. Later on I will show you some changes also, it is coming. See here, the last sentence actually, low dietary levels of folate, methionine, selenium can affect methylation. Research shows such defects are associated with neural tube defects, cancer and atherosclerosis. See here, it is a very important thing. Then we will go to these sentences. CpG sites are concentrated either in CpG islands located in approximately 60% of the human gene promoters or in regions of large repetitive sequences. For this you should know the molecular biology, what are centromeres, retrotransposon elements and all that. I am not going to describe them. You should know what are retrotransposons and centromeres and all that. So, unmethylated clusters of CpG pairs are located in tissue specific genes and in certain essential housekeeping genes. What are housekeeping genes? The housekeeping genes are those which do not generally, their expression do not change with the different conditions. They have to be required all the time. That is why they are involved in routine maintenance. So, those are called housekeeping genes. So, these unmethylated clusters are usually located in such genes. So, those which are active, they have, this is very easy to understand. When you have methylation, there is regulation of gene expression. Here you do not need regulation of gene expression, because they are going to be there. Their expression is not changing. So, they are unmethylated. So, when you require some change, you must have changes in methylation. So, that is why this is happening. We are guessing, because it is an observation. There are specific proteins that bind to unmethylated CpGs and initiate gene transcription. Remember, I told you there are proteins which recognize methylated nucleotides. So, similarly here also unmethylated CpGs can bind to specific proteins. This is how they remain in complex. methylation, unmethylation are signals for binding. Similarly, phosphorylation, if it is not phosphorylated, so those conditions, these changes ultimately are responsible for the protein-protein interactions. So, this understanding is very important. And methyl groups are acquired through diet, I already told, and are donated to DNA through folate and methionine pathways, with S-adenosyl methionine being the donor of methyl groups covalently attached to the cytosine. So, just not to leave out anything, I read that, but otherwise I have already told you. Thus, imbalance in dietary nutrients such as folate, vitamin B12 and all that can cause hypomethylation. This may lead to improper gene expression and ultimately the genomic instability. So, this happens, genomic instability is often seen during chromosome rearrangements. So, a common polymorphism in the methylene tetrahydrofolate reductase, the enzyme I just mentioned, increases the risk of breast cancer up to 3-fold in premenopausal women. This is one observation or one example where you can say that methylation plays an important role and the diet also contributes. That is why I am insisting on that dietary part. What are the consequences of this methylation or hypomethylation or hypermethylation? So, repetitive genomic sequences already we told are heavily methylated. DNA methylation protects chromosomal integrity by preventing chromosomal instability, translocation and gene disruption. So, this is one important contribution. Genomic imprinting, I guess you know what is genomic imprinting, also requires DNA hypermethylation at one of the two parental alleles in order to establish monoallelic expression. See, what happens is sometimes the X may be inactivated, that happens through this process. 2X are there in female, right, 2X chromosomes, one of them may be inactivated, that happens through this. Loss of imprinting is a mechanism of gene activation in some types of cancer also. Gene dosage reduction is observed in see here X chromosome inactivation in females, that is what I just mentioned. DNA methylation is needed for the germline specific expression. So, basically it is required for gene expression. Methylation is a mechanism for silencing tissue specific genes in cell types in which they should not be expressed. So, in other words, I already told you gene expression is a very important mechanism, because that only distinguishes liver cell as a liver cell, kidney cell as a kidney cell. Otherwise, the genetic information is there in all the tissues. What distinguishes them is their pattern of gene expression. So, if you have a regulatory mechanism that controls the gene expression, that is very important. That is why this microRNAs, this epigenetic silencing are very important. Previously, it was thought that the genetic changes are the most important ones. Now, we know this epigenetic silencing is also very important, especially with, as I told you repeatedly, tumor suppressor gene loss of expression is mainly through epigenetic silencing. So, that is why it is very important. There is an overall decrease globally in levels of DNA methylation in cancers. The loss in methylation is evident with repeat sequences, promoters of certain genes and in pericentromeric regions of the chromosomes. These are all some observations. These changes are linked with abnormal gene expression and chromosomal instability, which are all common in cancers. Promoter DNA hypermethylation is observed more frequently in cancers than mutations, especially for tumor suppressor genes. This is important since it is a known mechanism for this is a kind of repetition. The genes affected include those involved in cell addition, for example, cadherin, DNA repair mechanism, We will discuss that later. MLH1, cell migration, TIMs are tissue. What is TIMP? You know, tissue inhibitors of metalloproteases, differentiation, TGF beta receptor, and so on. So, there are many functional, what it means that methylation changes affect cell addition, DNA repair, cell migration and differentiation. Many functional consequences are there. Naturally, these are some examples where you see hypermethylated genes. BRCA1, this is so called breast cancer associated gene in breast and ovary. In cervix, P16 is hypermethylated, and kidney, TIM3, TIMP I told you, tissue inhibitor of metalloproteases and all, matrix metalloproteases usually. Then lung, P16 and P73, prostate cancer, BRCA2, pancreas, APC is again adenomatous polyposis coli, you know, I told you about this in colon cancer, APC, but in pancreatic cancer also. Uterus, MLH1 is again, we will talk about these genes later. This is another important thing. Interaction between DNA methylation and chromatin changes. H3K9 methylation and the histone methyltransferases are required for DNA methylation even in lower organisms such as Rhabdosis and Neurospora. There are also what are known as polycomb group of proteins which also target gene repression. For example, they have given H3K27 ME, ME means methylation, 27th amino acid lysine at the 27th position have been implicated in targeting and maintenance of DNA methylation. There are also methyl cytosine binding proteins, and the protein complexes in which they reside can bind to methylated CpG sites. This is also a silencing signal. Why we are studying? Because these are important in gene silencing. The complexes also contain HDACs. Here we are talking about acetylation deacetylation, which catalyze deacetylation such as H3K9 that are highly characteristic of transcriptionally silent regions. These are some examples. I think we will kind of stop here. We will continue in the next class.